Right now, literally as we speak, there is a high-stakes, microscopic war happening all around us. And honestly, frequently right inside our own bodies. We completely take them for granted when we pick them up at the pharmacy, but the science of how antibiotics actually work is nothing short of miraculous. So in this explainer, we're going to break it all down. The strategies, the specific weapons, and the incredibly intense evolutionary arms race of human medicine. Let's get into it.
So let's think about a massive paradox for a second. How exactly do drugs kill a pathogen without, well, killing you? Think about it. If we introduce a literally toxic chemical into your system to destroy an invading bacteria, how does the medicine know who the enemy is? A drug has to achieve something called selective toxicity. If it's not highly selective, the cure suddenly becomes just as dangerous as the disease itself.
And that brings us to this brilliant concept called the disease triad, which perfectly illustrates our incredibly delicate rules of engagement. Picture yourself at the top there. You're the host. Down on the left is the pathogen. There's this constant dynamic interaction happening, right? The pathogen causes disease while your immune system fights back. But the second you introduce a drug into the mix, things get super complicated. The drug acts on the pathogen, sure, but it can also act on the host, causing side effects. You have to metabolize the drug, all while the pathogen is actively mutating to resist it. It's a massive, incredibly complex balancing act.
Okay, quick terminology check before we go any further, because words definitely matter here. We toss around the word antibiotic all the time, but its true definition in biology is incredibly specific. A true antibiotic is actually a chemical substance produced by another microorganism. We are talking about literal biological warfare derived right from nature. Usually, it's soil bacteria that evolved these specific chemicals over millions of years just to fight each other, for territory. And that's actually totally distinct from the broader term antimicrobials, which covers the whole shebang: synthetic compounds, antivirals, and antifungals.
All right, so once we deploy these weapons onto the battlefield, what actually happens? Well, we basically have two main strategies: bacteriostatic and bactericidal. You can think of bacteriostatic as basically freezing the enemy's reinforcements. It completely halts their growth and replication, keeping the population static just long enough for your own immune system to step in and mop them up. Bactericidal, on the other hand, that is the obliteration strategy. Those agents actively eradicate and completely kill the viable bacterial cells.
Let's actually step into the lab for a second to see how scientists measure this stuff. They use something called the broth dilution method to find the MIC, or the minimum inhibitory concentration. It's exactly what it sounds like, the absolute lowest concentration of the drug that prevents any visible growth. Look at the tubes with no drug, they are super cloudy, right? Just packed with bacteria. But as you increase the dose of the antibiotic, the broth eventually becomes completely clear. That exact threshold right there, that's the MIC. The precise baseline needed to freeze the enemy's growth.
But wait, while the MIC tells us how to halt visible growth, what if we want to actually eradicate the enemy? That's where the minimum bacterial concentration, or MBC, comes in, and it goes a massive step further. The MBC is the lowest concentration that causes a 99.9% decline in the actual number of bacteria. See, if we take samples from those totally clear test tubes we just looked at and put them on a petri dish, we might actually still see a few colonies grow. That means some bacteria survived, just in a dormant state. But the MBC, that's the specific threshold where practically nothing grows back. It's undeniable proof of 99.9% eradication. Now, you're probably wondering, if we have drugs that eradicate 99.9% of bacteria, why don't doctors just prescribe the strongest drug every single time? Well, that brings us to narrow versus broad-spectrum antibiotics. A broad-spectrum drug is basically like carpet bombing. We use it when a patient is really sick and we don't know exactly what infection they have. But the risk is huge. It causes massive collateral damage, wiping out your body's good bacteria, your microbiome, and it heavily promotes pathogen resistance. A narrow-spectrum drug, though, is our precision strike. It targets very specific species, which completely minimizes the fallout. And the only reason a precision strike even works comes entirely down to the enemy's physical armor. Bacteria generally fall into two main camps: gram-positive and gram-negative. Gram-positive bacteria have this thick, highly exposed layer of peptidoglycan. It's basically a big, vulnerable wall. But gram-negative bacteria, they have a much thinner layer that is completely hidden underneath an incredibly tough outer membrane filled with these tiny pores called porins. So a narrow-spectrum drug might be perfectly designed to destroy that thick gram-positive wall, but if you fire it at a gram-negative bacteria, it'll just bounce right off that outer membrane. The weapon absolutely has to match the specific vulnerability. So what specific weapons do we actually have in our arsenal?
Well, we basically have 5 fundamental ways to completely dismantle the enemy's biological machinery. Number 1, we inhibit cell wall synthesis, stop them from building their armor. Number 2, we affect cell membrane permeability, literally punching holes in their defenses. Number 3, we inhibit their metabolic processes to just starve their synthetic pathways. Number 4, inhibit nucleic acid synthesis, which destroys their actual DNA. And finally, number 5, we target their ribosomes to completely shut down their ability to manufacture vital proteins.
And when you actually map those 5 targets onto the bacterial cell, the specialization is just incredible. Beta-lactams, like your classic penicillins, specifically hunt the cell wall. Polymyxins attack the plasma membrane. Fluoroquinolones dive super deep into the cell to interfere with DNA synthesis, while drugs like macrolides specifically bind to the ribosomes to halt protein production. Every single drug class has a very specific set of coordinates it's programmed to strike.
But hey, just knowing how the drug works in a petri dish isn't enough to win the war. When a doctor prescribes a treatment, they have to act like a literal general planning a battle strategy. They're weighing all sorts of clinical factors. Efficacy, like can the drug actually physically reach the site of the infection? Pharmacokinetics, can it maintain that lethal concentration in the body for long enough? Host toxicity, is this going to compromise the patient's own immune response? And combination therapies, if we throw 2 drugs at the problem, will they be synergistic and actually help each other, or will they accidentally be antagonistic and cancel each other out?
All right, here is where the story gets really urgent. The bacteria, they don't just sit there and take the hits. They launch a massive counterattack through resistance. They are constantly adapting to our antimicrobials, and they do it in 4 incredibly sneaky ways. First, spontaneous mutations make individual bacteria naturally resistant. Second is transduction, where these crazy viruses called phages actually carry resistant DNA between different bacteria. Third is transformation. This is where bacteria literally scavenge floating resistant DNA from their environment. And finally, conjugation, which is direct cell-to-cell contact where they pass resistance genes directly to their neighbors. They are actively, constantly sharing blueprints for survival.
Which leaves us facing a pretty provocative reality. As these bacteria rapidly mutate, seamlessly alter their cell permeability, create brand new enzymes just to destroy our drugs, and continuously share their blueprints for survival with each other, is our current arsenal going to be enough to win the next microscopic war? This evolutionary arms race isn't history. It is happening in real time in hospitals and communities all over the world right now. And it's exactly why understanding and protecting these absolutely crucial medical weapons is more important today than literally ever before.